Thanks. All right, thank you very much. Hello, everyone. My name is Jason Wittenbach. I work at the Genelia Research Campus at HHMI. So I thought I'd start by showing you a little picture of our idyllic setting that we have. Howard Hughes Medical Institute funds basic biomedical research at campuses and universities across the US. But at HHMI Genelia, it's a dedicated campus for studying neuroscience. And so those of us there are trying to use and develop modern tools to understand the circuits and mechanisms that make the brain function. I'm part of the Freeman Lab, so this is our fearless leader, Jeremy Freeman, there at the top. The rest of us, we're all some combination of computational neuroscientists slash coders slash maybe neuroscientists in our free time. So I'm going to tell you a little bit about some tools that we've been using or been developing based on Spark to try to understand the neural circuits and the data that we're looking at. All right, so I want to tell you about an experiment that one of our collaborators does. This data comes from the ARNS lab, and in particular from a postdoc, Yumu, in that lab. So what you're seeing here is a larval zebrafish, or at least a cartoon representation thereof, that's in a little virtual reality machine. So this is kind of our version of the Oculus Rift for a zebrafish. But if you ever get an invitation to be in the beta for a VR from a neuroscientist, I would recommend declining that after you're going to hear about what happens to this guy. Anyway, so the fish is mounted so that it can't move, and we have electrodes going into the nerves that go to the muscles in its tail that send commands to swim. So the fish is paralyzed while this is all happening, but the signals still transmit to the muscles so that we can infer when the fish is trying to swim. And to close the virtual reality loop, what we have is a small image projected underneath the fish such that when we detect the intent to swim, we move the image underneath with some gain function to simulate swimming with the fish. At the same time, the piece of technological innovation that the ARNS lab kind of pioneered is light sheet microscopy. So at the same time, they have a laser down there shining on the fish's brain. And these aren't just any fish. They're genetically engineered fish that have two very special properties. The first one is that they've been engineered so that the tissue in their brain is completely transparent to visual light. So that means that we can shine a laser in and it gets all the way to the neurons, and then any light that comes out we can collect as well. The second thing that's special about them is that their neurons have been genetically engineered such that only when they're active do they fluoresce. So only the neurons that are active will absorb the laser light, and then we put a microscope from the top to collect the output, and we can effectively observe which neurons are active at which times by collecting those light signals. So here's what the data looks like. Let's see, can you guys see that? Hopefully it's not too washed out. So the main figure at the top, you're looking down on the fish, and the top left is one of its eyes, and the bottom left is the other eye. His head's to the left and his tail's to the right, and these little glowing spots are the fluorescent light coming from the zebrafish's brain while this experiment's going on. At the top, you see a side-on picture, and at the left is a head-on picture. So we can collect data from the roughly 100,000 neurons that make up the brain of this zebrafish about 3 or 4 times every second, which kind of puts us on the forefront of data being collected from any nervous system anywhere in the animal kingdom. And so our question that we want to answer is, what's going on? Can we make some sense of all of these bright flashing patterns that we see? All right, so what does this data look like? We can sample about, the fish will stay alive in this experimental prep for about an hour or so, and at the sampling rates that we get, that gives us tens of thousands of data points. And then with the resolution of our microscopes, we can get a spatial resolution of tens of millions of voxels throughout the fish's brain. So if you put that all together, you get a data set that looks like a 4-dimensional array, t by x by y by z, that's roughly a terabyte or so. And so this isn't huge data, but in science, it's very important to be able to iterate quickly on experiments. When you do your experiment and you come up with a great analysis, an idea that you want to test, you want to be able to get those results back quickly so that you can iterate and do something new and exciting again. You don't want to have to spin this up on your laptop or your desktop and then wait a day or two for the answers to come back. And so this is kind of what we've been working on, is how can we leverage Spark to do general processing of n-dimensional arrays. And so I just wanted to spend a second and talk about arrays. Most of you probably know about these from various and sundry programming languages, but I think a lot of times in the data science world, we're used to just two-dimensional things, right? A data table, which might be n rows by k columns. And that's great, that's a little two-dimensional array, but our data has a little more specialized structure in a couple ways. The first way is that in a general data table, your k dimension can be heterogeneous, right? One column might be integers, the next column might be strings. For us, every single column and every single entry in every row is exactly the same data type, usually when it's coming straight off the microscope, some kind of unsigned integer. However, as we've seen in these experiments, right, we can image a single plane in the zebrafish's brain and get a three-dimensional array. We can also do the full three-dimensional volume by scanning our laser up and down, and that gives us a four-dimensional array. And some of the most recent experiments that we've seen coming out of our collaborators' labs now interleave two different colors of lasers that activate neurons in different subsets so that they can identify one group of neurons versus another by the color channel that is coming out of. So as you can imagine, I think our data arrays can get arbitrarily large. And so these are the kind of data that we're working with. And so just to continue the example a little bit, I want to show you what some of the raw data looks like, and we'll get a little bit of an idea of what kind of analyses we'd like to do. So again, I apologize for the contrast not being great here, but this is what the raw data is from a single plane in one of these fish. And hopefully what you can see is that you might see a little bit of a glimmer from what looks like a neuron, but in general, there's lots of background noise. So the first thing, to make those nice videos like we showed you, we need to do a lot of image processing. And once we do that, right, we can get down to some nice videos like this, where you can actually see the individual neurons apart from the background. And to do this, when we think about doing this in a distributed setting, it's useful to think about how we're going to represent our data, or how we're going to think about our data. So the first way to think about it is kind of how it comes off of the microscope. And this is as a collection of images, one saved out to each file, and then they're indexed by time. And so this is really useful if you want to do some kind of image processing, like apply a sharpening filter or do some downsampling. You can simply load each image into a single record in an RDD and then map to your heart's content to do image processing. However, on the other hand, we often want to kind of think about an individual pixel in one of these images and think about how its value changes over time, right? Time series analysis. And when we do that, it might be more useful to think about our image, or to think about our data set as a collection of time series that are now indexed by space, right? And so if you're using an n-dimensional array in something like NumPy in Python, this is as simple as a transpose, right? Are you thinking about your array as x by y by t, or do you want to think about it as kind of t by x by y? And that's really simple when you're not distributed, but when you are working in a distributed environment, the nuances of how to handle this get a little complicated, right? And so being able to change back and forth between each of these in our data sets is really important. For example, to make those images we showed, we want to do basic image processing, thinking about our data distributed as it is on the left, and we want to do some kind of time series processing as you see it on the right. And this is a very general thing. So this example is from neuroscience, but all branches of science can benefit from this, right? So pretty much anyone that collects data that comes in this very structured form, from radio telescopes in astronomy to satellite images and geospatial data analysis to climate change being spaced by time as well. So we previously developed a bunch of very specific tools to neuroscience, but we kind of wanted to generalize so that people in other domains could make use of these tools as well. So this leads us to Bolt, which is the Python package that we've been working on. So Bolt is a distributed ndarray. It's built on PySpark because a lot of people in the scientific community already use Python to do their scientific computing. There's a great ecosystem there and a lot of tools, and so we wanted to be able to leverage that, which made PySpark a nice choice. And then also in the Python ecosystem, many of you are probably aware of NumPy, which is a great local ndarray implementation that many people are familiar with. And so we wanted to build a distributed object that conformed to the exact same API to kind of minimize the overhead in switching from maybe a scientist who's done a lot of stuff on their laptop and now they want to leverage a cluster computing environment to analyze their data. All right, so this is the kind of thing that we're shooting for, right? If data is some n-dimensional array, we want to be able to do things like take the mean of a single axis across a single axis. We want to be able to take a transpose or do things like indexing and slicing to select interesting pieces of our data that we might be interested in. So we had to make one big API choice at the beginning. Or I guess it's more of an API/implementation choice at the beginning. And the choice we made was to introduce this idea that we called the split. And so we decided that in our bolt arrays, as we called them, that we would have only one extra piece of metadata that the user needed to think about. And that was a particular location within the set of dimensions, such that all dimensions before that location are going to be distributed across your cluster, whereas all dimensions after that will be kept together as a NumPy array within a single record. So if we go back to our example, right, of a series of images versus an image of series, you can see that in the left, we want to think about our data as a bunch of local XYZs, right, kept together in a single array on an RDD, and then we'll just have a label for time, since that comes to the left of the split, right, to keep track of that. Whereas in our image of series representation, you're going to have a transpose, right, such that the label now is the location in space, and then we'll keep each time series together as well. So this is what this kind of looks like at the RDD level. So if you think about what one record in one of these bolt arrays looks like, let's take, as an example, just a generic 5-dimensional array. And if we decide to put the split after the second index, that means that each record will keep track of your W and V indices, just with a little tuple, and then we'll actually use a NumPy array, since this is in Python, to keep the rest of the data local on that particular record. And so if we move the split over by 1, right, what that involves now is having 3 elements in your tuple in a 2-dimensional array in each record. Or you could get even crazier and move it 1 to the left. Then you have a single index, and I'm still working on my art skills for drawing a 4-dimensional array, but I think it looks something like that. All right, so what different methods do we expose? How do people interact with this? So the first set of functions or methods that we put on the bolt array are things that are familiar to anyone who's used NumPy before. So we allow you to do things like indexing and slicing, applying functions along axes, and all of these things act exactly like they do in NumPy with no changes. And then there's a second set of operations that have equivalents in NumPy, but now because of this idea of having a split that separates your distributed from your local indices, these operations take on a bit of a new meaning. So now things like doing transpose actually involve shuffling your data around the cluster, perhaps, right, if you end up switching a distributed index for a local index. And then we added on a bunch of other things just because we like the functional programming style that things like Spark use. So we added on a bunch of functional primitives as well, and then a couple of extra things such as chunking and padding, which are just little goodies that I'll describe later that only make sense in a distributed setting. All right, so what do these different things look like, just to give you an idea? So doing something like indexing, right? Let's say we had a 4-dimensional array with 2 distributed indices and 2 local indices. Well, if you index into one of the distributed indices, right, one of them before the split, then that's just a filter. You go through all of your records and you throw away the ones that aren't included in the index or the slice. Whereas if you do your indexing or slicing on one of the last 2 dimensions, then it becomes a map, and you just go in and actually index or slice into every local array in an equivalent fashion. Doing something like applying a function along an axis can be kind of simple or tricky. If the axis that you want to apply along is one of the local dimensions, then it's easy. It's just a map. You go in and you apply that function to the appropriate axis on each of your small arrays in the records. However, if you want to apply the function along one of the distributed axes, then it can get a little tricky. Sometimes we can do something clever, like a reduce, if the function that you want to apply is a mean or some kind of aggregation. But otherwise, this is where a lot of the work that we've done has gone on to get kind of fast and efficient implementations of these things. And then the interesting one, the transpose, right? Since we have this idea of a split separating your local and your distributed axes, the transpose is kind of a very important thing, as it determines how your data is laid out. It determines what kind of operations will be efficient and what kind of operations might take a while. So if you do a transpose, and all you'd ask me to do is transpose 2 of the indices that come before the split, well, then that's really easy. That's just a map. I go in and I flip all of those indices in each record. And the same thing if you ask me to do it on 2 of the indices that are both local to each record. I just go in and flip that as well. Where it gets tricky is if you ask me to flip one of the if you ask me to transpose a local index with a distributed index. I won't go into the details. There's a lot of bookkeeping, but it effectively ends up being that there's a shuffle that goes on, and this is kind of the big expensive operation that comes that you want to limit in your workflows. And so we've tried to optimize this pretty heavily. We wrote our own custom partitioners for this. Since this data is highly structured, we know exactly how many records there should be, exactly where we want them to be partitioned. So we've tried to optimize that as much as possible. Right, and so this is just reminding you now that if you do have one of these workflows like I was talking about, you want to do some image processing first, and then maybe you want to do some time series processing later. Well, what you do is you would load your data in this form with the split after the first index, do a bunch of apply along the first axis, which is going to be cheap, and then you can do the transpose, which is going to shuffle your data, and then you can do your time series processing afterward. All right, so one thing I wanted to mention that I said I'd get to is this idea of chunking, which is kind of an extra feature that doesn't exist in a local NumPy array, but we found to be really useful in a distributed array. So take as an example this kind of time series of images data set. If your individual images, as in our case, are very, very large, you might not be able to get the desired level of parallelism that you need to get the most out of your cluster. Right? So let's say you only have 1,000 images, but you have more than 1,000 CPUs that you can work with. You're going to be underutilizing your cluster if you don't repartition. But in our current framework, there's no way to get any higher level of parallelism out. And so we introduced this idea of chunking, which is you can take your local arrays and then define a block size on each of those, and we'll chop the arrays up. And then what we'll do is we'll separate each of those into its own record, and underneath the hood, we'll keep track of extra indices that tell you which chunk you're in. So what this allows you to do is really fine-tune the level of parallelism in your workflow if you're an aficionado, right, and want to put that time and effort in to really get the maximum out of your cluster. So just a couple examples of how chunking is pretty useful. A lot of times the first thing that we want to do when we're analyzing this data is ask, can we go in and actually find where the neurons are in the brain? So this is a source extraction problem. Each volume's pretty big, and so it might be nice if we could parallelize across our volumes. So if you think of this as a two-dimensional array in X and Y, you could chunk your array up into a bunch of little pieces and then run some kind of source extraction algorithm on each of the individual pieces to try to find the neurons. Which is great, except for you have a little bit of an issue, right, where some of the neurons, like in this kind of top left corner, seem to be spanning multiple chunks, and you need some way to deal with that. So we've also added in the feature to have padded blocks, right? So when you chunk your data set up, you break it into partitions, but then you include a little bit of the data from nearby pieces in your array. So you do get some data duplication. It's inefficient in that sense. But what it allows you to do now is to apply operations on a chunk-by-chunk basis, which hopefully drastically reduces the size of your data set. Then maybe afterwards you can collect the results of where these ROIs, these regions of interest are. And then, so this is what you get if you apply the algorithm without the padding. Now, if you apply the padding, it finds lots more ROIs because there's extra neurons in each bit. And then what you can do is you can collect afterward and then stitch the results back together across the different chunks. And you get an output like this. The second thing that chunking is really useful for, and as I alluded to, is for tuning the level of parallelism in your cluster or in your job. And so being able to we've tried to make some optimizations underneath the hood to choose nice chunk sizes for our users based on what would be a good level of parallelism before we do shuffles in transposes to make sure that we're not extremely under-parallelizing or over-parallelizing the data. All right, so just to kind of wrap it up, I mentioned at the beginning one thing that was really important to us was to make the API for this very similar and almost identical to the API for the NumPy array. And so some of you may have heard of a package called Thunder that our group had developed recently, a couple of years ago, which was very specialized for neuroscience data. And so what we've now done is we've kind of torn out the guts of Thunder and replaced it to be built with both bolt arrays or NumPy arrays in mind. And now since they both have the same API, we can write one piece of code that works both locally on a single machine or distributed across a cluster. And this is nice, right? Because when you're doing data analysis, you might want to prototype an analysis locally on your laptop on a small data set before you move it to full production on a server. And so we're excited to see what kind of other specialized tools people might build based on this kind of unified API. All right, so to wrap up, I'd like to thank everyone in the Freeman Lab who's helped build this. Also thank the people at Genelia Scientific Computing who have been wonderful in terms of all of our demands to get this up and running on their cluster. All these projects that I talked about are open source and in GitHub repositories, so if anyone's interested in using them or contributing pull requests, we're always happy to have more people. Thank you for your attention, and I'll be happy to take any questions.
That was fascinating. I can't wait for NumPy to analyze my neurons and my behavior. But anyways, if you have any questions for it, I think you guys are just doing an incredible job. I've heard a couple of speakers from the Freeman Lab. Fascinating. Anybody? Right. We're going to go in the backward time series now. Can you keep your question short?
I was wondering, as you try to structure your data, how that interacts with if you want to do like linear algebra operations on a distributed array. Do you have to worry about which direction you want to transpose things in order for those algorithms to be efficient, or did you implement your own versions of linear algebra or those kind of things?
Yeah, so that's a good question. We've implemented a few versions of our own thing. There's actually there's another open source project that we've talked to some of the developers from called Sparkit-Learn, who have been kind of trying to do this. And they were interested in knowing, could you take something like this bolt array and make it a drop-in replacement for the NumPy array and doing machine learning things? It gets a little tricky. Some of that's because we made this decision to have the split, right? And how you transpose your data makes a big difference. We're definitely interested in pursuing that. For now, for the analyses that we're really keen on doing, we've written our own versions of them. But yeah, we're definitely interested in figuring out how we can generalize that to be as useful as possible to as many people.
Looks amazing. Sort of a two-part question, I suppose. One, is the underlying implementation actually in Python, or did you write a NumPy-compatible API layer to a Scala implementation for the actual processing? And the other one was just, what were your final results? How large of a cluster are you running this on, and how much of a difference did it make in the eventual process that you're doing?
Yeah, so to answer the first question, it's all written in pure Python. So far, we've been able to see good performance results with that, and so we've stuck to that level. Usually at Genelia, we have our own cluster, so we've been running it on usually somewhere in the 10 to 20 nodes, each of those nodes being 16 CPUs with a quarter terabyte of RAM on them, and you get huge speed ups for some things, right? So like, for example, loading the images and just applying image filtering to each one, it's almost exactly linear scaling, right? Other than the overhead of getting your cluster set up. So for some workflows like that, it's hugely beneficial. Once you start shuffling the data, then it gets a little tricky. But what we found is that a lot of things that would take kind of, run it overnight on a single machine, we can now do in an interactive session instead.
Great talk. So following up with the linear algebra idea, say like standard Hadoop or MapReduce type of structure where you would only have one dimension, you could be limited by if that one dimension is overly large, then you couldn't fit everything on it. And to take a bit of an example, say if you're trying to do some sort of convolution or calculation like that, what you might actually care about is the 2 or 3 dimensional local structure, which looks like you could fake is the wrong word, implement with how you do the chunks. So what I'm curious of, with those chunks that you showed, in that example I saw, it kind of looked like the chunks were working on their own. Is there any way to do sort of matrix multiplication, that sort of thing, across chunks so that you can get maybe several more dimensions that you might need for the operations, and not across the entire dimension of each, across the entire length of each?
Yeah, that's a great question. Right now, we don't have anything specific implemented for that, but you're exactly right. I think the chunks and the padding would be the big thing there, right? So let's say, hopefully I understand your question, right? If you have a bunch of time series and you want to apply some convolutional filter across each and every one of them, but each individual time series might be too big to fit in memory, you could chunk it and pad it, and as long as your pads are bigger than the window of your filter, right, then you still could technically reconstruct based on that. So that's the kind of approach I would take for that. We haven't, like I said, we haven't built anything specific, but that was exactly what we had in mind with allowing chunking to hopefully facilitate workflows like that.
Anybody else?
Way in the back.
Oh.
Hi. Since we're in Python, how does Bolt compare to the Dask array?
That's a great question. So we've actually been talking a lot with Matt Rocklin, who you may know, who works on Dask. So when I mention here, right, that we've used Bolt and NumPy to kind of abstract away local versus distributed on Thunder, we've also talked with the Dask people about having a Dask backend for Thunder as well. So if anyone in here is not familiar with Dask, Dask is kind of for out-of-core computing, right? You have one machine, the data's too big to fit into RAM. How can you load it in to RAM in pieces in a way that's kind of sensible? And they've recently also started a project called Dask Distributed, which might be a little bit of a competitor to Spark, who knows, but to use Dask across multiple machines. And so we've been benchmarking with them. Right now, we're trying to figure out how to get both of our workflows into the same EC2 cluster so that we can compare them directly. So far, it looks like we're a little bit faster than Dask, just in terms of being able to flip the arrays, right? That transpose, the shuffle is kind of the big operation. Yeah, but we're definitely interested in doing some more comparison there, because I think what would be great is if you could add Dask to the list of things that Thunder abstracts on top of, right? So if you have a really small data set, use NumPy mode to run it on your laptop. If you have a big data set, but only a single beefy node, you could switch to Dask to do out-of-core computing on one node. And then if you have a cluster at your command, you could switch to Bolt to do full distributed.